whole oxygen
I bet
signaling
those
cascade
potatoes
is.
are high too.
Had to be at the.
Yes, obviously.
So.
It was great chicken nuggets.
Yeah.
Just. Yeah.
Oh, okay, I just wanna make sure mine wasn't
Very-
broken.
Yeah.
You get to.
Really don't.
This,
Like this.
I don't necessarily.
And I like. They don't like. She's like, I know. I'm aware. I.
I do my thesis.
Which Betty the Taco Bell.
Yeah, you know what it is, yeah. Sky Bridge to get there, and then you just chill. We're in there.
Pretend you're at Taco Bell here.
Somebody in a.
It doesn't have. Back when all the fast food you have like. Back that late.
Yes, yes. Oh, it wasn't. The day.
Back when Pizza Hut was like a Pizza Hut. How could be anyone? There hasn't.
Oh, now it's just, now it looks like.
It might be a stretch, but.
It too. They changed. I also had, I've heard of the chandelier. Chandelier. I am like the husband's my actually borrowing machine. I feel like it's changed.
What is going on? The one,
It's like we're making. You gotta. I was when I was little. Front wheel, yeah.
I'm like, I just want.
Set it up.
No, no. Yeah. Yeah.
It didn't really taste
Chocolate milk, that was.
and then everything really smelled.
Yeah. Yeah, yeah, this is, I thought I had done it right, but I actually. Ratios for the last question, I was like, oh, I forgot 0.7 times the credit, but I had like 1 grader. Interesting. In the lab, I don't leave milk, but. What part of your hair? Well, this is very. It's like McDonald's, you should have the $2 chicken.
So.
I don't know which way, but I don't like it.
Way,
Yeah. Yeah.
They don't have to do it. You just find them.
It's like, so bad. But.
Right about the church.
Just.
Can't beat that, you can't relax.
Yeah, it wasn't for.
Like as soon as I hit high school, I was like.
That, there's no steps.
Yeah.
Environmental cleaning, it's all.
Oh, so.
We're not a charity.
Also has
So
to
they
say
order
it was
like. Waiting on down the date.
Yeah. Yeah, they're like ours.
They're like. It's something.
See, the other day, they said I agree with Rich cigarettes. Probably. Rich cigarette.
I feel like that's a common thing I've heard of.
Yeah.
Once we have.
I don't. Never mind.
Ton.
So.
Yeah, it's.
They have like fantastic.
Like get the hype around. Maybe we have things.
Maybe a bigger.
Yeah, going to.
And they also I feel like people who shop there are not used to that.
If not, compared to the
Stay away.
of whatever else is out there. Yeah, I also, yeah. Yes.
For everything else.
Yeah. They're like, oh, this is creme butter.
We're for seminar by Francis Chan, who is seminar today, and he's going for some the training process this year. So we're very excited to hear updates from Francis. I'm going to give a very brief introduction because not only I'm really excited to hear what Francis has to say, but looking at his CV makes me want to get to work. It's also very hard to summarize, but just some brief highlights for the person that I have. Francis got his bachelor's at Hampshire College in 1993 and a PhD from Cornell University College of Evolution. He joined OSU as a postdoc in 2001. And after being a postdoc, he moved into the assistant professor senior research rank and moved up the ranks there to 2022 when we were able to hire him as in that line as associate professor. So he's now looking at the tenure aspect of that process. That happened in 2022 until now. And since 2021, so 2020, Francis has been the director for CIMMERS, Cooperative Institute for Marine Resources Studies. Big agency integrating research in different areas of very good work there. Overall, Francis has been mentor to 7 to 11 students here. A lot of co-advising early on. Now has been able to have a lot of students and over 65 peer publications very impressive and a lot. The amount of money that Francis been able to bring was OPI and PI needed has been pretty astounding. Over $29 million across per year, over 10 million in the last 6 years. So something to, again, want to go back to work soon. And I'll say that one thing that's really impressive about having known last 10 years I've been here and also now seeing CV is the amount of integration that his work has been science level, but social justice, stakeholders and legislation, all that stuff is the most impressive type of central science that I've seen. And also in terms of mentorship of students of different backgrounds, he has definitely put the money where his mouth is in terms of recruiting and providing support from students of all kinds of backgrounds. And lots of collaborations with indigenous groups and tribal leaders, things like that. So I definitely recommend learning from Francis about how to be a better, how to use your science for bettering society. So we're gonna go ahead and get started right now.
Right. Thank you everyone for coming this afternoon. It's really exciting to be giving this talk for tenure. I'm going to talk about the history of oxygen, but it's going to be a very brief history. But of course, there's a really, really cool, deep history of oxygen and evolution on Earth. If you're a biogeochemist, you might think about the frenemy relationship between rubisco, which is the most abundant enzyme on Earth, and oxygen. It evolved during a time when oxygen is already very low, and it's little that consequence ever since. So it's because of photorespiration, it's an oxygenase, which I will come back to later. It actually is a very inefficient enzyme. But the deep story of oxygen has something for almost everyone in biology. The Cambrian explosion is part of the story. As oxygen rose, you can have large animals. And also a story about extinction, great extinction events such as the Permian extinction. And kind of my favorite is when insects are the size of cats, because oxygen was 35% of content in the atmosphere. And I think there's even a snake thing about their the hemoglobin being very high affinity because of their demand for oxygen in subterranean habitat. So something for everyone. But I would talk about that. I would talk about a recent history of oxygen and really through the lens of my own research. And it's going to be, and I hope to impress upon you that there is a pretty interesting recent history that relates to climate ecosystem dynamics of oxygen. That and there's a story about novel cogs and the biogeochemical cycling of oxygen and other elements that we perhaps have not appreciated. And of course, from a lot of what I do nowadays, it's actually just a story about the science of climate solutions that originates right out of OSU. And I'll start because this is a little atypical because my path is probably not the same as most other associate professors. I want to just give a brief summary. As Felipe noted, I came in 2001. I had the opportunity to join, a lab of some of the premier marine ecologists on the planet, which is amazing. So Bruce and Jane, and I stayed on. And during this time, I've been able to work with students, postdocs, oftentimes in co-advising mode. And in 2022, I transitioned to tenure track. And I think you can see from here that was really transformative. It really accelerated my training and my research as a faculty. I was happy that I got out of this basin of attraction where I was relatively not cited and unproductive. And thank God, being an administrator doesn't seem to have to slow down my relevance of science. And if anything, I've been taking on more and more research as a PI. So this is money that is where I'm the lead PI. There's because I've been part of big team science, that number is higher. But again, I want to just acknowledge just the team science that I've been a part of. For tenure talk, I should be talking about what's happened in a very defined period, and that starts at 2019 for my clock. But I also want to go a little back further in time, just so that you have some context of, what is it that I've done and what have impacts and why am I doing certain kinds of research today? And I'll try to be relatively brief about that. When I came to OSU, this was really the state of climate science. If you were to ask a room full of marine scientists, what are the top 3 concerns about climate change that we had? One, the sea level rise. That was sensible. Talk about that a lot. Number 2, the sea level rise. And number 3 was sea level rise. We didn't really have that ecosystem perspective on what climate change would do to the ocean. I remember sitting in a panel with environmental law and science panel, and all the lawyers were talking about coastal zone management and sea level, and all the scientists were like, there's a lot of other things that are happening that you guys might not be aware of. But fast forward to today, and this is the most recent IPCC report, Ocean and the Cryosphere. We see that we have a really much more nuanced and complex picture of how climate will affect ocean ecosystems. Yes, we still have sea level rise somewhere. Where is sea level rise? Oh, there it is. But we also have things like marine heat waves, changes in pH ocean acidification, and oxygen needed into the mix. What changed? What drove this? And I just want to emphasize, I'm going to talk a little bit about how we went as a community, science that suggested that it's not just sea level rise to actual policy change. But I am not the only one responsible for it. I am a small piece of that conversation, but I want to walk you through that. And for us, it started with an early work where we found that there was evidence that that the ecosystems that is in our backyard, these upwelling ecosystems, they were actually the oxygen inventory was actually sensitive to climate variability. In other words, ocean oxygen inventory, which I measured, might not be as boring as we thought. You might think, hey, oxygen, how's it gonna change? But actually it was quite dynamic and with real ecological and economic consequences. And we were able to publish this finding in Nature, which I think started to help people coalesce their thinking about, oxygen and climate. Maybe there are some connections that are worth exploring. This was not settled by any means. Actually we're in a lot of science conversations where, they thought this was pure BS. But a couple years later, another paper came out in Science that was really influential. And this was the first record evidence-based understanding that the ocean was losing oxygen. And this is a term that is coined ocean deoxygenation, which tells you that nobody with any marketing skills talented. But this became a phenomenon that the ocean was losing oxygen as a consequence of climate change. And actually, a couple months before that, we had published our own paper highlighting that the system in our backyard was subject to this very rapid intensification of hypoxia stress, and actually where we were seeing anoxia, zero oxygen conditions that were unprecedented in the modern instrumental record of the ocean here. So I think some of this science like this helped to establish the rise of ocean deoxygenation as a mode of climate change. And then our own research has been trying to understand, well, is that sensible? Should we expect the coastal ocean, which is so dynamic, the temperature is not an average temperature, the winds are shifting. Should we expect our local ecosystem to really be coherently responding to these global forcings? We've shown that the ocean deoxygenation, in that the offshore water masses are changing in oxygen content. That's the water that comes up on the shelf. But the question has been, because we are an upwelling system where winds from the north is the engine that drives ocean currents. And those ocean currents are really important because it pulls deep, low oxygen, but also nutrient-rich water onto the continental shelf, where it eventually sees the light of day. And I think, and I also want to emphasize that this is a timeline, so that water comes up and it moves offshore and it ages. And this story about aging is going to come back later on in my talk. But the question we had was, well, do winds really matter for ocean dynamics? Because in a windier ocean, they a more oxygenated ocean after all? And are they actually changing? And actually, you just have to make the measurements and yeah, it really matters. And what I've plotted here on the top are these are winds from the south to the north, and these are winds from these are winds from the north to the south, and these are winds to the south, which are upwelling favorable winds. So every time we have these blues, they're upwelling favorable, and every time we see those, we see that they're corresponding to the decline in oxygen because we're pulling up that deeper water that is low in oxygen. And depending on where you are in kind of shelf, sometimes you're pulling water that's really, really low in oxygen. So there is this mechanistic connection between wind forcing that we see and oxygen that we can measure. And I want to mention that this is part of a mooring program that is now going on 23 years. It's the longest continuous record of mooring observations that we have of oxygen on the West Coast. And somewhat surprisingly to me, upwelling wind stress has also been intensifying, significantly so. I'll confess that when I first started, I think I looked at this record early on and I thought, oh my God, wind forcing is so variable. Because we don't really know what next summer is going to be. I didn't expect that we would emerge from that background variability to have a significant trend of increased upwelling in my so far in my career. And that this change, this enhancement of coastal upwelling has contributed to an intensification of hypoxia across the Northern California current. This is work led by my colleague Jack Barth. This is a project that I'm the lead PI on. And we reconstructed oxygen habits of Jack did, the oxygen distribution spatially across the Northern California current from the first 3 decades of observations into the middle and then into the 2020s. And you see that 2 things is that blue is bad and it's gotten worse. But there's some geography that it repeats. So there are areas that seem to be persistently sensitive to hypoxia, and these are, for the most part, we can explain that. We can explain from physical oceanography why certain places are hotspot. Okay. I would be remiss if I did not also mention that, let's say you don't care at all about oxygen. It's not interesting to you, but all you care about is acidification. Well, it matters because ocean is deoxygenation is linked to ocean acidification. And we see this from our instrumented record, where on the bottom in the blue is dissolved oxygen. And you see it moving up and down consistent with temperature because we're seeing water masses coming in and out. But that picture is mirrored by pH that we have installed both on sensors mooring right offshore to those that are right in intertidal. So when we have low oxygen stress, we also have acidification stress. I'm doing something that my physical chemist colleagues don't like. Is you need 2 parameters of the carbonate system in order to estimate other parameters. But for the waters that we have given our alkaline saline temperature, about 7.7 is the cutoff for dissolution. So things below 7.7 tends to be where if you're building a aragonite shell, that will dissolve in that water mass. And we have it here. So, and you can actually, not right now, but a month ago, you could have gone to see and actually taken organisms that these shells are dissolving in our coast right now. So that's the biogeochemical and physical context. But Harris, what does it mean? Nobody eats oxygen. Nobody goes to the coast and orders a plate of oxygen. It's about food, right? And the question that we've had is, well, how low is too low? I mean, we've been somewhat arbitrary and said, as a field, we've been somewhat arbitrary by defining what thresholds are for oxygen impairment. A common one, actually, I found out refers to when shrimpers stop catching shrimp in the Gulf of Mexico. There's not a generalized biological reason why there's one cutoff, but we really weren't sure what it looked like for organisms on our coast, because maybe they're fine because they've been here for quite a while. And this is work that my master's student Hannah conducted where she looked at early life stages, red abalone and Dungeness crab. And we've been investing quite a bit in kind of apoptosis-based technology, to measure oxygen, certainly with respiration. Felipe does that as well. And what she finds is that, of course, we don't, I mean, these are aerobes, we know that at some point they will die or their respiration will decline. But we didn't know where that cutoff was. Was it really close to zero? Was it far from zero? And in both cases for red abalone juvenile and Dungeness crab megalope, we find that they're actually quite, it's pretty far away from zero that they go from where an organism where the respiration is oxygen independent to a point where their respiration rate is dependent on the amount of oxygen in the system. So imagine if your metabolism is dependent on the amount of oxygen in the room, well, there might be spas and change your metabolism. A second thing that we were looking at this work was really, we wanted to know at what point does phenotypic variance collapse, where there's at some point where nobody does well, and we're interested in what that point was. Now, this is the news from 2 days ago. We're unfortunately in the midst of another heat wave in the Northeast Pacific Ocean. It's remarkable how often this is happening. This is where I was going to joke. So George Somero, who was a faculty in the department, I was chatting to him one time. We were on a panel together, and I said, George, he really made us understand the temperature dependence of metabolic processes. And I said, we're looking at 1 degree warming. What's a degree amongst friends, George? And then he, of course, he goes on and educates me about the importance of one degree. But we're looking at 4 degrees. So we're actually looking at some pretty large changes in the environment that these temperate organisms that we work on live. But is there a way to, should that matter that we're having low oxygen and warming? And is there a generalizable way that we can look at that? A collaborator of mine, Curtis Deutsch, but although this is not working on, he developed something called the metabolic index about a decade ago now. And he argued that you can look at the performance of an organism if you knew a couple of things. Well, one, you want to know what, of course, the environment looks like. So this is PO2, the partial pressure of oxygen. If you know something about the oxygen sensitivity, that P crit, at what point do they go from oxygen independent metabolism to oxygen dependent metabolism? And then you need 2 other things. One is a biomass scaling, which he tries to argue doesn't matter because he's a modeler. He tries to make everything not matter. So he gives reduce the first. And there's the radius function, right? Because there's a temperature scale. Well, we do PO2 and oxygen sensitivity, and my student Nyesha, who is in Australia right now fighting mud crabs, she is actually this is part of her research going into the future. She's trying to find these terms for a number of different crab tags. Like if you like Nyesha, she loves crabs. She found a project where she's able to really ask, is sensitivity to this ocean stressor, can it be predictable simply by relatedness or systematics? Or can you be predicted better by traits or by environment, so where they came from? Pretty important question, and I'm not sure we quite have the answer, so I'm excited for her to find out if she comes out of our fingers, of course. So where do we stand today? I'm showing you some science that really started my career, science that I've continued to carry forward. It's remarkable because I was in, I would be in AGU trying to argue that, no, this is a thing, people. And today, I can write in a fairly good journal that, this is really ocean epoxy. It is a science of climate change in the ocean. And we have the IUCN published a tome, and this is a tome. This thing was 560 pages. I authored one of the chapters, so I got a paper copy. This thing's thud. And you can see that, and this is to reinforce the point that I do not claim even small amount of credit, but just a little bit of credit for moving the field in this direction, because we are here, but this was a community effort. The science community did the work to get us to this point where we understand what these different modes of climate change impacts might be. Although, I like to measure impact also in terms of have we changed people's minds about this? This is early on, these are early days, and I was excited. The New York Times writer came out of us and he wrote an article, and it was that dead zones creates alarm and skepticism. And the quote from one of the fishermen is that, they just go on and on about global warming. And there was a lot of mistrust, right? We were not connecting with the coast community. Fast forward to 2025. This is Crystal Adams. She's the executive director of the Oregon Dungeness Crab Mission. We needed defenders for science today. We needed third-party validators that can articulate why marine science is important. And I asked her if she would appear in the video. I also asked her if she would provide a quote to support a legislation that's pending. And she gladly did it because she understood just how important science is and that the realities of environmental change, climate change that her industry faces. So we got somewhere. Impacts. Ooh. Okay, I'm going to switch gears. So that was the first chapter. I'm going to talk about the middle chapter, and where I like to convince you that there are some interesting new cogs in the biogeochemical cycle of oxygen that we perhaps overlooked. And it starts with this question. So it might sound odd, ironic, because I've spent my career telling people hypoxia and anoxia is a thing. Be careful, be careful, be careful. I felt like a bit like an ambulance chaser. But I've made about I think somewhere around 1 million measurements of oxygen in the ocean. And when I look at those 1 million records, I'm like, I don't see a lot of zeros. Why is that? Why don't, we have a system that's always teetering on the edge of hypoxia, if not anoxia. And it's really rare that I actually see a zero. Now that was just the gut feeling. So I looked at the data and I plotted the data. I worked in the Northern California Current, but I took data from the Central California Current, but also the Southern California Current. And this is a cumulative frequency diagram. And what you can see is that there's this odd hinge that there's a lot of zero. There's not a lot of zero observation. There's not a lot of observation of zero oxygen concentration. And that observations really take off once you get past this hinge of about 25 micromolar. And for an ecosystem ecologist, I look at that, there's a negative feedback, so that as the system approaches a very low oxygen value, there must be a mechanism that is acting that slows down that approach. Now that's typically how we think about biological systems or biogeochemical systems. But there's the real question: is that a stupid question? And I've explained why this should be a stupid question. It's this guy. Negative feedback control when we're at 25 micromolar doesn't make sense, because this is textbook understanding that established by Louis Pasteur, Pasteur, that microbes don't really care about variation in oxygen concentration until you get to at least 2 micromole, and this is for facultative anaerobes, where they will switch from aerobic to anaerobic metabolism. And it's also textbook that cytochrome oxidases, those terminal enzymes in the respiratory electron transfer chain, they have an exceptional affinity, nanomolar scale affinity for oxygen, dissolved oxygen. And at the same time, you should know in the field over the past 15 years or so, us biogeochemistry geeks, we got a new hammer. We got a stop sensor. So this is a sensor that allows you to very, very precisely measure nanomolar dissolved oxygen. So this was 3 orders of magnitude more precise than we used to be able to. Actually, I think my group was the first to deploy this in the US West Coast. We were collaborating with a colleague who invented this. It's really elegant sensor. So fragile. If you looked at it wrong, it would break. But we got a new hammer. So the whole field began chasing dynamics of exceedingly low oxygen environment, so they would, expeditions would service all over it, they would go off the coast of Chile and Peru, looking at these suboxic, genoxic systems. And so the whole field was running towards the very, very low concentration kinetics and dynamics. Oh, okay, I should take a step back and I want to just explain what I mean by low, by infinity for dissolved oxygen. And if you're at Mark's talk, this should be very familiar to you. This is a functional response, right? That, you know, there's some substrate, in this case, oxygen, and there's a rate of dissolved oxygen use. The more substrate you have, the faster the rate of use, until asymptotes to something called Vmax. And half that Vmax, the concentration where you can have the Vmax is something that we call Km. You can also think of it as prey and attack rate. Mark, yes. But the canonical understanding is that, I mean, dogma, literally dogma, is that respiration has this incredibly low Km, very high affinity. You can grab oxygen like nobody's business. And this is a cartoon because I can actually, you know, have a nanomolar and micromolar curves on the same page. But it should be that nothing interesting should happen on the flat part of the curve. Because there's no biological rationale for anything changing. So when you're above nanomolar concentration, nothing should matter. And then I was talking with my colleague Steve Giovanni, who's actually very close by, and he said, "Well, what about all these other enzymes upstream of that terminal oxidase?" And so there's all these oxidases that they catalyze that incorporation of oxygen into compounds, they oxidize compounds, usually free of energy. Sometimes they're doing it to make things. You know, I think when we teach biology, we talk about this part, right? Glycolysis, pyruvate, citric acid cycle, phosphorylation. But there's also this other part where upstream, you take something that's really complicated or very stable, like tyrosine, an aromatic ring. A lot has to happen to it before that terminal oxidase can use that the energy that's locked up in those carbon bonds. And along the way, someone's got to keep breaking it until it gets to a point that's usable. And Steve said, well, some of these oxygenases, he said it's known that they have higher Kms. And I was like, okay, great, maybe that's a thing. And then he also says, oh, we just published a paper that these monooxygenases, they're really, really highly expressed in microbes, especially in the deep ocean, which suggested that like microbes actually need these toolkits in order to access carbon. And then I did what should have been an uninteresting experiment was that I work in one of the most productive ocean biomes on the planet. You cannot see your hand once you dip in the water. I mean, it's so productive. There's no reason that microbes should be limited by organic carbon because they're living in an organic carbon soup. But I did this experiment where I added C6 glucose versus control, and every single time the community exhibited very, very profound organic carbon limitation. So that didn't make sense, and it's what we also realized that they're using up everything that's laid out very quickly. So what's next? They must be dependent on oxidases that allows them to access all the other carbon that's not as easy to assimilate. So this led us to develop a hypoxia barrier hypothesis. This is HPH1. And the hypothesis states that the low oxygen affinity of oxygenases upstream in that metabolic chain acts as a negative feedback, which as biogeochemists we like, that limits the actual distribution of N-oxy in the modern ocean. And if this hypothesis is attractive to us because it posits 4 predictions at different scales of organization. At the molecular scale, we expect oxygenases and terminal oxidases to differ in their Km. At the organism scale, so individual microbes, their Kms should differ depending on whether they're respiring complex carbon, so the stuff, the aromatic rings, versus simple carbon like glucose. At the community scale, we expect that once you run out of the really easy to eat stuff, rates of respiration are gonna be limited by the ability of dissolved oxygen at micromolar, so 10 to the negative 6, rather than nanomolar, so 10 to the negative 9 concentration. And then the ecosystem scale, we should expect that, you know, that should manifest as a buildup of hypoxic water masses that just fails to launch, it fails to progress into anoxia. All right, so we have 4 predictions. Let's see, we're at different scales of biological organization. Let's see how they fare. One was this was probably the most OCD moment in my career. I looked at, I don't know, this is not my literature, but I looked at thousands of papers on enzyme kinetics. And I pulled out about 1,000 that had oxygen KMs, and I organized them versus I split them between cytochrome oxidases and all these other oxygenases that are known to cleave really odd compounds. And there is a clear separation. What is the magnitude separation between the KM between the 2 kinds of enzymes? On top of that, also OCD moment, I looked at cultures where people looked at microbes growing on, you know, glucose versus, you know, toluene or other compounds. And it turns out that depending on what they're growing on, they have very different oxygen kinetics. So molecular and organismal scale, that it seems it could be consistent HPH. Also, it turns out people have been doing experiments that I was trying to do without knowing it for a while. We did go to see, we went to the oxygen nosomal here, and we did very simple control experiments. We added oxygen before we capped it and let it run. But it turns out a lot of people that work in low oxygen environments, because it's so hard to get a rate when oxygen is very low, they almost always add a little oxygen into the water, into the sample in order to get a rate. It's also sometimes it's really hard to really get a very, very low oxygen sample to fill up. And so people have been doing these experiments, and in every instance, we find that the parent KMs are not nanomolar. There's always a functional response where there's interesting dynamics in the micromolar concentration. And then number 4, we find that this is work with my colleague Curtis Deutsch, who's at Princeton now. You know, he's a great global modeler, and he actually interrogated both the model and the data set. And he finds that, interestingly, in the global ocean, there's missing volume of water that should be very low oxygen. You see this very strongly in the Eastern Tropical Pacific, which is the largest OMZ that we have, that there's a huge buildup of low oxygen water, and they fail to transition to anoxia. Same for the California Current, and for the Atlantic Ocean. So we're seeing evidence that, you know, there's a missing cohort of water that never made it to anoxia. And I should also mention this bracket is, if you talk to the modelers that do climate models, this is the range of KM values that they use. So they might be using the right value, but not for the right reason. And you ask them, like, why do you use that? They're like, well, because otherwise the model blows up by very low KM as a microbiologist. Okay. So those are largely correlative evidence. We really wanted to test this, do this experimentally. And this student that Steve and I co-advised, Sarah Wolf, and also an undergrad that was working very closely with her, you know, they went out, let's create a phytoplankton bloom in the lab, and let's put the water and manipulate the oxygen availability. So we're pretty good at manipulating gases, so we can set up this kind of ghetto time machine where we go back to low oxygen condition or today's modern oxygen condition. And in this experiment, we actually shifted up after a certain amount of time. So this sets up a battle royale between, you know, our grad student and Louis Pasteur. And interestingly, and I want you to look at focus on these rising curves. So this is the amount of oxygen that's used in each treatment. The green is the fully oxic treatment, and the orange brown is the hypoxic treatment. Remember, nothing important should happen in that flat part of the curve, but we're finding that that's not the flat part of the curve. And so what we found from these experiments was that, yes, community respiration was reduced by micromolar concentrations of oxygen, consistent with our hypothesis. But there are two things that were really weird. One was that when we upshifted, we increased oxygen, the prediction was that this orange curve would actually rise and meet up with this green curve because there's been this buildup of carbon that hasn't been used. That didn't happen. So whatever was processed during those hypoxic conditions remained unusable. And then the third thing that was really weird was that total organic carbon, which is this curve, it did not decline in concert of oxygen utilization. You notice that while it declines initially, it kind of flattens out, even though oxygen continues to be used. This is really weird. Because so the ratio of oxygen used to carbon mineralized, that's the respiration quotient. It should be around about 0.7 if you take into account various compounds. But we're finding that it's not a constant value and it can be very low. Just to give you context, so I've looked at, I think, every paper that have actually measured respiration quotient in aquatic microbes. There's about maybe 24. But you will find probably 5,000 or 10,000 papers that assumes a respiration quotient of 1. But in those papers that actually measured it, they're all over the place. They're really variable. So this ratio that we've been comfortable assuming across biology, we might be wrong. And we only get it because, I measure oxygen really well. Our collaborator does Craig Carlson, he's the best in the business for TOC. My student Haley Hudson right now, she has an experiment going, and we're going to add a third component, which is measuring of the inorganic carbon to test if we're right that respiration quotient has been overly assumed. But that's the story from the microbe side. But what about the carbon side? What's happening there? What's happening to this kind of flat side curve? And why do compounds that were processed under hypoxia, why do they appear to be recalcitrant even when oxygen is raised? This is what led us to propose HBH2, that there's a class of organic compounds that can be characterized by their oxygen dependence in use by microbes. And these compounds are more recalcitrant to degradation. Now, to get at this, and this is, I want to emphasize, this is, I have to let it go because I don't get to do everything in a project. This is work that a grad student that Steve and I co-advised, Ben Daniels, is leading on, along with Daniel Petras, a colleague at UC Versailles, who's an expert in computational mass spectrometry. And when you have that kind of hammer, you get to really interrogate the actual organic compound that are in your experiment. This is something that was accessible to me for a long time. And this is a Van Crevelen plot looking at the stoichiometry of oxygen, carbon, and hydrocarbon. But the key thing to pay attention is that the light blue is what was termed labile, things that just was really rapidly disappearing. The pink is this oxygen-dependent organic compound, and the purple is recalcitrant, things that did not change in concentration at all. And the expectation is that, hey, you know what? It shouldn't be different. It turns out is the labile and the oxygen-dependent DOM, they are different, but the oxygen-dependent DOM are just like the recalcitrant DOM, their signature. This is an aromaticity index. This is the nominal oxidation state. And Ben really it's a very creative and elegant study. And then he went to the natural system because there's a building literature. It's by no means as big as genomes, but there's a building literature on chemical compounds, and he sees that in natural systems, this OD, oxygen-dependent DOM, is chemically like recalcitrant dissolved organic carbon. You're like, oh my God, why do you care about this RDOC, recalcitrant dissolved organic carbon? So there are a couple of ways that we sequester carbon in the ocean. One is, there's the solubility pump, and this is why we worry with the, what if the jet stream, really warms up and we stop sinking water in the North Atlantic, then we're losing a huge solubility carbon sink. But there's the biological pump. It's kind of intuitive, things, grow and then they die and they sink. They carry with the carbon that they made the seafloor. But there's another important carbon pump, and it's called the microbial carbon pump. And in this formulation, the concept is that microbes, it's not an ashes to ashes, dust to dust world out there. Every time microbes process a bout of reproduction, they leave something over it that is kind of no one wants to eat, and that stuff is really recalcitrant. And that stuff has half-life in about 10,000 years. This is stuff that turns over, that stays around a lot. So every time microbes chew through organic matter, including nutrients, they make it a little more refractory. This is important because this RVOC pool is about the size of the atmospheric CO2. So pretty big, really long half-life. It's driven by microbes, and this manipulation of the microbial carbon pump is it has emerged as a key climate mitigation tool. If we were somewhere else, we would be talking about this. We just had a scientist from China come visit. He's the author of the microbial carbon pump concept, and he let loose that he's been given $100 million to work on this. So others are on this case. But our results suggest that there's an existence of a novel oxygen-dependent microbial carbon pump. Although we won't see any of that $100 million because the FBI said, no, we can't work with that. Okay, so there are 118 elements. You might be wondering, oh my God, you hired somebody that works on one, maybe two. So I'm gonna give you one more, and this is nitrogen, and this really is inspired by my earliest observations here as a ecologist, where I go to the coast and just look at productivity of the ocean, as it is incredible. And you look across the global ocean, it's highly variable. It really changes a lot. And the question I have is like, well, what's driving it? Is it simply that you have more nutrients, or is it the efficiency in which nutrients are used? Does that explain the structure, ocean patterns of productivity? And I mentioned that nutrient use efficiency, nitrogen is one of nutrient, of course. It has a pretty deep basis in our field. So the seminal work by Eugene Odum really was one of the key pieces of key paper that really got us thinking about nutrient use efficiency as a property of ecosystems. It's always amazing, like just back when you could just inductively reason and make a science paper. I don't think those days are with us anymore. But that concept, we see it today in ecology. It's used to explain, for example, the biodiversity function debate. Is used to look at plant invasion in systems. And recently it's been looked at metabolic scaling theory. So it's something that is persistent in our field. And I just ask, well, what is it possible that efficiency plays a role? And by efficiency, I define it by the proportion of ecosystem nitrogen that is been appropriated by phytoplankton so that they can double in size growth. And should we expect a system where, as you have less and less nutrient, that in a sense the prime producers they overdo it, that they're compensating? And this makes sense, right? Because if you were to think, well, in the tropical system, in coral systems, very nutrient poor, there must be all these compensatory mechanisms that allow organisms to more tightly recycle nutrients and more efficiently use those nutrients. So that makes really strong biological sense. And you would not be wrong if you made that assumption. Or does scarcity act as an ultimate constraint, that there's just no way around the problem of nutrient limitation? And I did this by looking at the literature. This is the meta-analysis. And it turns out that harm reduction really scales predictably with the amount of ecosystem N that you have. It's actually not, it's non-linear. This is a log scale, and you can see the exponent. So it's a highly non-linear relationship. And surprisingly, contrary to what we as ecologists would expect, the places with the highest nutrient use efficiency is not the lithotrophic ocean, but it's the most productive ocean that we have. A little more nerdy, but that the variation that we see in chlorophyll is about 4 orders of magnitude. The variation that we see in the size of ecosystem N pool is about 1 order of magnitude. That's important in a second. So why is that? Why do we see this pattern? And we propose essentially an ecosystem ocean succession model. Remember I said, okay, pay attention to this time, that water comes up, the light of day, it's the youngest water on the planet. And then it moves offshore, it becomes part of tropical gyre, which are some of the oldest surface water in the ocean. And this is we're arguing this is really a time axis that as the system ages, a couple of things happen. Phytoplankton become a less dominant part of the biomass. You start to lose nitrogen out system, and then most of the system becomes dominated by organic, dissolved organic nitrogen. And then functionally, what it means is that early in the system, you have high export efficiency because things are in particles that they can sink. That as you switch to a more dissolved pool, well, that dissolved organic nitrogen doesn't sink, so the retention efficiency is very high. So that acts as a feedback mechanism that constrains. So there's that you now know why I like negative feedbacks, right? And there's a mechanism by which we never run out of nutrient in the surface ocean because the process that stimulates that loss slows down as you get a small pool. But on the other end, you can't build it up continuously because you increase the loss term. And when we look at the data, we see, actually, as the system becomes more and more oligotrophic, it becomes more and more dominated by dissolved organic nitrogen. Okay, this is the home stretch. And this is some work that a lot of work that I've been really focused on is like, can we translate climate science and climate solutions? This is part of our university strategic plan, climate change and solutions. It's not just about telling people how bad things are, but are there something that we do about it? And here's a challenge from a crabber that I spoke with. He said, "Where should I go tomorrow to get out of this dead zone?" He just went out full of his pots. He had 4 live crabs, and it was not a fish store because there was a fisheries and wildlife observer on board. I said, I don't know where I don't know where to send you because we actually don't have a lot of observations. For moorings that are real-time reporting, because I don't want to tell them, oh, you know what, a month ago, you should have gone here. We need real-time data, and we have very few assets out there. And unfortunately, 6 of them are going to blink out this winter because of the NSF's termination of the OI operational funds. So we're turning off the headlights. And geography is really expensive. I play in oceanography, and it's really expensive. It's not just gas money. But could we crowdsource ocean observation? We have a fleet. We have a very extensive fleet off the Oregon coast. You look at AIS, the vessel tracker. There's somebody out there doing something all the time. Yeah, we just need to give fishermen one of these things. Not super realistic. And I just want to make a plug. There's a ship time opportunity that's coming up. We used it to provide experiential learning opportunity. This is Brooke from, I think she was an IB student. So in 2 days, she was able to really learn how to completely man a CTD rosette and tell the captain when to stop. But these are things that we can't just give to a fisherman. So I've been exploring a co-production approach. What if we were to provide crabbers with low cost, high performance, smart and rugged EO sensors? And this is highly collaborative. Starts with Jeremy Childress, who was a student at OSU. Actually, he worked for me. Now I'm one of his clients. This was his master's thesis sensor. It worked, but the crab fishermen there's no room for crab. It's small. And so we had a grant and he made a smaller version of, more rugged. It's the same probe that are on gliders and power floats. And we're able to give this to fishermen, so now they have a tool to know, when they're fishing in a low oxygen zone. And really, literally overnight, we increased the number of monitoring stations that we have to dissolve oxygen. So that we can start to identify, where are those hot spots and refuges in our state. And I should say that it's not just the West Coast, this is actually a lobsterman Cape Cod. So the company that built this for us also was starting to sell this because Cape Cod Bay is having, now they have a epoxy problem. This is Haley up in Puget Sound, where she's been working with the Lit Up tribe. They've been interested in not only their crab fishery dynamics, but also they wanted data to be ready to sue King County for violation of the Clean Water Act and their treaty rights. So different use for data than I had imagined. But we also, this is my other master's student behind us. This is an opportunity for us to ask some science questions, and I think this is actually really important for us. It used to be that we did a lot of basic science, and sometimes it's used by somebody somewhere. The funding landscape is completely shifted. We're much more likely to be funded to do something that is straight up problem solving. So it's really up to us. How do we continue to sustain some of the fundamental science that we want to do? Here we're asking, what's the value of additional information? So, you just had upwelling winds is free. It's easy. You have winds and temperature, also free. What if you had in situ measurement? This is because the dynamics are quite redshifted, that we can have fairly good 1 day to 2 day predictability of what oxygen should be, which might matter for fishermen so that you can say, look, okay, pull your pot out today. I wouldn't leave there any longer. And we can also start to get at what's the geography ability because we just have so many sensors out there. There's also another aspect, which is, what's the strategy for dealing with non-stationarity? And non-stationarity is kind of like a $10 word for saying, we might not know what we're talking about very soon. And this is a really good example. This is work by Alan Shanks, Dungeness crab megalith, so the young of the year versus the commercial crab catch. When he first started it, it was this regression without this X. But as he's progressed, these are heat wave years. The system is really changing faster than this that he's building in increasing power his statistical relationship. So how do we deal with what changes ahead? We need to go just double down on mechanistic model. We're going to build the ultra uber mechanistic model that takes us from genes to organism, or we can do completely AI-based correlative models. I'm trying a different approach. It's like, well, what if we had a good indicator that is reliable, such as year 3 crabs? Of course, it turns out nobody wants to count a whole bunch of year 3 crabs, but working with technology partners, we're able to, we've developed these sensors and they're going to go out in the state coast-wide fishery next month, where they're going to help us measure a whole bunch of crabs that no one ever wants to measure. This is work that my student Emily is leading on as part of her work, trying to understand kind of temporal dynamics and ecology. Trent Wallace, who's undergrad in my lab, is also helping. And Amari and Naisha helped build some of the training data set that we've used. I should also mention that for the fleet, they want to do this because it turns out in order for them to get sustainable seafood certification, they need a way to forewarn of population collapse. This is not a fishery that is assessed. It doesn't make sense to have it's a complete overpopulation, so it doesn't make sense to have assessment because you can count every single female to be up to date. There is no predictability of how many individuals come back next year or 4 years later. Turns out having this tool might help them get sustainable seafood certification, which will allow them to sell to the EU. Okay. I am going to actually skip the next part, which is about a multi-stressor project that I'm working on. And I'll just say that, we have big challenges. And when we look at this, like somebody should work on this. It's like, well, who? It's us. we're the marine scientists, we should be working on this, but it's really hard. And I've also come to accept that I can't do it all alone and that I need a team to do this kind of science. And it's a really awkward thing because the scientist is like, okay, I'm gonna assemble a team and then I'm gonna let them do the work. And I do have this team, I'm leading a 17 PI effort. We gave us a little over $4 million to make it work. And we're working with tribal fishermen, resource managers. I'm gonna go to the end. I want to say something about my other job. So I do have two jobs. I run this is something called Cooperative Institute for Marine Ecosystem Research Studies. I have about 24 employees that work that I supervise. We do about the research portfolio about $40 million. And we're part of the US National Cooperative Institute Network, which together we're about $500 million a year Earth System enterprise. And my job is to make sure that OSU has a seat at that table. We do things ranging from marine ecosystems, conservation science, ocean acoustics, Earth-ocean interactions. You might say, you don't do most of this stuff. And it is true, but I've learned that I have to write proposals that fund this stuff. So I have to get good enough. And it's not just SIMRS people that benefit from it. Over time, we've grown a number of outside SIMRS faculty, students that are funded by research grants. We're going to be renewed for another 5 years coming up for hopefully another chunk of money. But this is my most treasured outcome is that by being a director, I get to retask money. And I have a business plan where I'm able to tax NOAA, tax my own science, and turn that into support for students. So in the past 4 years, I've made available just a little more than $400,000 direct support for students, students of color who participate in good science and work with fantastic researchers that we have at OSU. I won't tell you more about what's ahead. You saw a lot of things that even though I spoke about them, a lot of who helped me, that we were part of teams. If your name is not up here, I'm sorry, but you should be. And I'll leave it, I'll end with this picture and thanks for the Chan Lab for being there. I grew from a party of one, to, a party of many, and it's just been exciting and I really treasure it. Well, thank you.
Time for one or two questions?
David.
So the climate's changing, the earth is changing. We heard about this first in the Gulf of Mexico, and then it became an Oregon thing. Where is it happening next? Hypoxia specifically.
Well, Cape Cod, so East Coast.
Yep, that's a new one to me.
Actually, for a different set of reason, China, and it's because of really continental scale eutrophication. We're seeing like hundreds of square miles of ocean become hypoxic. For us, it's been kind of small invaders, but we're seeing it in Southeast Asia, huge areas, and their ocean use is completely different. They're also thinking about, hey, do we use this to make more recalcitrant DOC to get carbon credits? So that there's a different science opportunity.
You know enough about the process now where you can predict where the next most vulnerable can be.
I've had some collaborators where we're trying to do a global synthesis, and we break it down to fundamental, like what are the 2 or 3 axes that explain all systems. And usually like the physicists argue like, no, it's the fusion term. Then the biochemist, no, it's the 7 term. We haven't gotten there yet. I don't know, it'll be interesting. Yeah, the next dead zone project.
I'm curious because you mentioned sort of this idea that you're interested in like predicting outcomes of settings, and you have all of these potential variables and all these complex interactions, stuff like that. Have you guys thought about like competing predictors directly and just seeing like what are, because I think that's, to me, that's a tricky thing where as you add more things, you might get something that looks better. Or you might actually just putting in a bunch of junk in there that's unnecessary. So how do you handle this thinking at this scale?
For which part of them?
For like any of it, I think you mentioned like, it's like it's kind of a central thing, right? Like you said, like, do you just use like some machine learning approach where you're just purely getting an outcome, or do you try to have something that's informed based on like functional knowledge?
Yeah. Guess my example is like if you're a high school principal or superintendent, like, okay, how many science teachers, 10th grade science teachers do I need? Now you can build an incredible model of, demography, macroeconomic trends, and try to forecast immigration of, this school district. We can call it middle school, principal, like, hey, how many 8th graders you got? So I'm looking for that. How many 8th graders you got?
Yeah, it's obviously like, how do you handle that? I don't know, like, how do you figure that out in these systems where it seems like you're characterizing a whole bunch of different things, but when are you evaluating what's the most important?
We haven't done that formal evaluation. That could be a really good.
I think building on that question too slightly, you had mentioned, and I might have missed what the reasoning is, but you said you were focusing in on year 3 crabs or like this indicator, why you're.
Because marine organisms, the mortality schedule, mortality is super high when you're really young. That's almost so that by the time you get to year 3, the natural rate of mortality should be relatively constant and certainly way smaller than it's experienced during the other parts. That's assuming that that's the mortality schedule. That should be right, because otherwise, crab should be like 8 cents each. Something right. Thank you so much.